Good evening, everybody, and thank you for coming for the first of this series of lectures. I'm sure you've already heard an awful lot about antibiotic resistance in the last year or two anyway on the television. It seems to be on all the time. Indeed, some of you may have seen me on the TV before now, so it's really nice to talk to people in the same room rather than through a little TV camera. So what I want to do is really give you quite a global overview about antibiotic resistance, and then really bring it a bit personal. So personal to me, what do we do in my research lab here at the University of Birmingham, so you can understand the sort of things that we do in research, and then make it go out again and sort of talk about the issues and why there are some real problems with antibiotic resistance at the moment, and what it means for us as individuals, as the general public. So I think it's really important we put it into context first. What is an antibiotic? So the word antibiotic comes from two Greek words, antibiosis, which means against life. And when it was first coined, it was used to mean any antimicrobial, against any microorganism. But nowadays, it's taken to mean an antibacterial drug. So it's become synonymous. Antibiotic is taken to be the same as antibacterial. And the purpose of today, every time I say antibiotic, I am only talking about drugs that work against bacteria. I'm not talking about drugs that work against viruses or fungi or any other microorganism. So where do antibiotics come from? They can be produced naturally by other microorganisms. They can also be produced by things such as plants. We can make them as well. Chemists are very good at making antibacterial compounds in the lab. So whether they're natural or whether they're made in a lab, they can be discovered or they can be invented. And wherever they come from, these are molecules that will either inhibit the growth of bacteria or they will kill them. So antibiotics don't always kill all known germs, with that well-recognised phrase. So let's have a quick show of hands now. How many of you have ever had an antibiotic? The banana-flavoured medicine, yeah? Okay. So, you know, that's how much we value these drugs. We've all taken them pretty much. So how do antibiotics benefit all of us? So this is just a timeline across the bottom here, from childbirth right the way through all of our lives. And in the pale pink, I'm showing the sort of infections that we all get today, but have existed for thousands of years. They've plagued mankind forever. So antibiotics have allowed many of us to survive to adulthood. Without them, we would have died. Without them, many of our mothers would have died soon after childbirth. However, going through into modern day medicine, much of what we do is only possible because of antibiotics. So for instance, surgery was really able to make the major advances because when patients got wound infections or bloodstream infections after the surgery, they were able to treat the patients. Because what used to happen is the patients would survive the surgery and then die of an infection. 21st century medicine is incredibly sophisticated. We do heart, lung transplants, liver transplants, we do knee replacements, we do hip replacements. We consider these very commonplace. But these are again only possible because of antibiotics, because many of these are done in an increasingly elderly population, and that's the time when the immune response tends to wane and we become more susceptible to infections. So antibiotics are hugely important to all of us throughout our lives. However, unlike other therapeutic areas of medicine, say drugs that we take as heart medicines or anti-cancer medicines, just to name a couple, we use antibiotics outside of human medicine. We use them in animals, we use them when we're growing vegetables, when we're growing plants. They're used hugely in food production. They're also used in brewing, in beekeeping. We use antibacterial paints on ships to stop them getting fouled up with slime. And of course, in labs such as mine, we use them when we're genetically manipulating bacteria. We use them as markers so we can trace what we're doing to the bacteria. Or people use them in genetic manipulation of food crops. And really, can you imagine now that we would do that with a cancer drug? If I replaced the word antibiotic and said, well, I'm going to use a cancer drug and paint it on the bottom of a ship, you'd actually start thinking, this is a really mad thing to do. But we do it, and we're completely flooding the planet with antibacterial compounds. We even use them in our homes. How many of you have used the blue washing up liquid or the blue hand wash? Hands up. That's an antibacterial compound that will select antibiotic resistant bacteria. So you think you're using it for the best possible reasons. I think we're using it for absolutely the wrong reasons. Soap and water, if you wash your hands properly, will almost certainly suffice in most situations. And I'm going to touch upon this several times now. So why do we value antibiotics so much? Well, these are the sort of mortality rates that were associated with dying from what started off as a skin infection or a wound infection. And you can see when two types of antibiotics, sulfonamides and penicillin, were first introduced, there was this massive plummeting in the numbers of people dying from these types of infections. Now, it's also true that people were starting to understand an awful lot about microbiology by that time, and hygiene was improving too. But in the pre-antibiotic era, as you can see, pre-1930, really huge numbers of people died. And this is just one example for you. So how much antibiotic are we using today? So this is a heat map of Europe, and cold colours show not very much antibiotic and hot colours show a lot. And this is just measuring the amount of antibiotic used in patients out in community, so they're not in hospital, but this is for intravenous antibiotics. Now, there is a caveat with this sort of information, that not all countries necessarily do a lot of this type of treatment. So in this country we tend to have more IV antibiotic use in hospitals, but even so, we're like most Northern European countries, we don't use a lot of antibiotics compared to some, such as the Southern European countries. And one of the reasons for this is the way our antibiotics are available. Nonetheless, we're still using more and more antibiotics. So this slide just shows increasing year on year amounts of our blockbuster super duper drugs, the ones that will kill all known germs. And the reason we're having to use these is because many of the other antibiotics that we would normally have used even 10 years ago are now much less effective because of resistance. So we're increasingly having to get out our reserve drugs, our special weapons. Now, why are some of those southern European countries and other countries outside of Europe, why do they use so much more than us? Well, because there is uncontrolled use of antibiotic. You all know that when you've had an antibiotic, your doctor has had to write a prescription for it. Without that prescription, you really would find it very difficult to get an antibiotic. In other countries, people can just go into their pharmacist or a market and they can just buy an antibiotic. And this picture here was taken by a friend of mine in Vietnam, and they were just buying the antibiotic that's shown on the right-hand side. And you can get antibiotics, whatever you want. The difference is that when a doctor gives you an antibiotic, they tell you to take it 3 times a day usually. Now, 3 times a day, to many of you here, that will mean breakfast, lunch and dinner, won't it? What it really means is every 8 hours. So even though we have controlled use in this country, we still don't always take them properly. And the reason we dose at those regular intervals is to make sure the levels in your body fluids are at a high enough concentration to inhibit or kill the bugs causing infection. And we also tell you to take the drugs for the entire time you're told to take them, 5 days, 7 days, 14 days. And if you don't do that, you're not going to get rid of all the bacteria causing the infection. So as soon as you stop taking them, they'll grow up again. So the length of time to take the drugs is really important. It's taken doctors 5 years at medical school and 4 to 10 years postgraduate training to get to the position where they know how to treat patients and diagnose infections and all the other things they have to be able to diagnose. I can go into a pharmacy in Greece or China and just buy an antibiotic. I don't need to know anything about it. I don't know how much to take, but I'm going to take some anyway because that's what I think I should be doing. Antibiotics are miracle drugs, they'll make me feel better. And it is absolutely the wrong thing to do. One should only be given an antibiotic or take an antibiotic if you know you've got a bacterial infection that is going to respond to the drug that you are taking. And that's one of the big issues in antimicrobials at the moment and is driving resistance. So what is the problem? So the problem is we've got an increasing number of infections by resistant bacteria, and unfortunately we have very few new treatments coming along. So until relatively recently, it didn't matter so much that we had antibiotic-resistant bacteria. They've always been around. I've luckily been able to spend my whole career working on antibiotic resistance. So it's kept me in business. The problem is that until recently, we could just go to the pharmacy and get another drug out. And many of the drugs were developed to be active against the resistant strains we found before. And because by understanding resistance, we can then use our drugs that we've got better. We can understand how to develop new drugs that aren't affected by the resistance. And we're just not making the new drugs at the moment. So there are at least 200 antibiotics worldwide that are available for patients to take. Obviously not all of those are available in the United Kingdom, but many of the ones that are available just aren't effective against some of the most serious infections we're now seeing, particularly bloodstream infections. And this is just an example from a hospital in the UK. So this ward had 27 patients on it, and 7 of them had bloodstream infections by E. coli. And that's a bug that normally lives in your tummy. It doesn't cause a problem at all, but if it gets into your bloodstream, it's in absolutely the wrong place and will have devastating consequences. Now, of those 7 patients, 6 of them were really so ill they were very near dying. So they're very severe invasive infections. And for 2 of those 6, the options for treating the patient were really restricted. On the one hand, because the adverse reactions that they would suffer from some of the drugs that they were still available, and on the other, because of the resistances. So this is a real problem affecting patients every day in hospitals throughout the world. Now, antibiotic resistance is not new, and Alexander Fleming, in his Nobel Prize-winning acceptance speech, said, "It is not difficult to make microbes resistant to penicillin. The time may come when penicillin can be bought by anyone in the shops. Then there is the danger that the ignorant man may easily underdose himself and by exposing his microbes to non-lethal quantities of the drug, make them resistant." What a prophecy, because that's exactly where we are today. So, if you look at this timeline, how long does it take to get antibiotic-resistant bacteria emerging? So if we look very carefully here, at the top here, this is when antibiotics were first available to be used in patients. And if we look at the bottom here, on the same timeline, it's when resistance first emerged. And you can see very clearly here the sulfonamides, penicillins, streptomycin, just by following the colour lines down, that very soon after introduction, you see also the emergence of resistant bacteria isolated from patients. So it occurs really quickly. So how do these bacteria become resistant, and how does it happen so quickly? It is a very, very good example of Darwinian evolution. So if we look on the left-hand side here of this slide, this could be a test tube, or it could be a human body, or an animal, or any other environment. So what we've got is a population of bacteria, millions of them, and amongst those millions could be a single or very rare bacterium, just happens to have had a mutation in it to allow it to survive a hostile environment. Now that hostile environment could be high temperature or low temperature. For today's purposes, it's antibiotic. And what happens is that over a very short amount of time, the antibiotic kills all the susceptible bacteria, leaving that resistant bug to grow up and become the dominant population. Now the generation time of E. coli is 20 minutes, which means within 24 hours we've got millions and millions of resistant bacteria that have emerged. We do this experiment every day in my lab because we're trying to really pressure the bacteria to become resistant. Then we do experiments to try and find out how they've changed to become resistant, so we can start trying to find drugs that are still effective or doses that are still effective. So how do we measure resistance? Well, on the top left-hand side of this slide is a little agar jelly plate, and on the surface of this we seed it with a bacterial suspension, and on top of that a little filter paper discs which have been impregnated with antibiotic. And where there's a zone of clearing around the little filter paper disc, you can see that that microorganism is susceptible to that antibiotic. And where there's no zone of clearing, that means that that antibiotic is not effective, the bacterium is resistant. Now we can do another test on the bottom right here, and that's called an E-test, and we've got strips of filter paper here, and instead of the antibiotic being in a single concentration, it's in a range of concentrations. And if we look here, going from the top of the strip where it says E down to the bottom, this goes from very high concentrations down to low, and there's actually numbers on this strip. And where this growth of bacteria, this clearing, crosses here, we read the number off, and this is the minimum inhibitory concentration to inhibit the growth of the bacteria, and we use this value. So what does this value mean? How do we use that to say the difference between sensitive and resistant? Well, there's something called the breakpoint concentration, and this is worked out with an equation based upon how much drug gets into a patient's serum, for instance, or their urine, or into their lung tissue, and how long it stays there. And for every antibiotic it's different. And then they base it upon a geographical distribution of bacteria, and will say a given value, say of 16 microgram per ml of drug, is concentrations higher than that is resistant, and below it is sensitive. And we use these as what we call cut-off values. So this is an example of a timeline of isolates from a patient that we've done a lot of work on. This patient had a salmonella infection, and you can see that this patient we were sampling over 20 weeks. The numbers, the L numbers, refer to the different isolates that we kept and we've researched on. And underneath it shows you when we've given different antibiotics. We gave this patient lots of antibiotics. Within the first few weeks of being exposed to antibiotic, we got multi-drug resistant salmonella. The patient had infections in the bone, in the bloodstream, and after 21 weeks this patient died. We could not treat this patient. This is an example of the type of problems that are being experienced now. So we went on and did some experiments with these strains to look and say, well, how have they changed? And these are some of these MIC values, and you can see that L10, one of the isolates that were obtained after a few weeks of treatment, is pretty much resistant to everything that we've got on this slide. So my lab tries to understand how the bacteria have changed. What's happened to make them so resistant? And we work on a pump system, which is basically like a vacuum cleaner. So when a drug gets into a cell, it vacuums it straight out again. So when we first started working on these isolates, we ruled out all the common things that had ever been described before, and we started then looking to see if there was this vacuum pump. And these are some of the results that we got. So on the left-hand side is a little graph of how much of the drug ciproxin, CIP, gets into the bacterial cell. And the vacuum pump is something which we call efflux. It's transporting out of the cell this drug. And in the presence of an inhibitor, it didn't really make much difference, and lots of drug was in the cell. But if you look at the resistant strain on the right, the blue line shows you that there was very little drug in the cell. But when you inhibited that vacuum cleaner, lots of drug was in the cell. So we knew straight away that we were dealing with a bacterium that was able to export lots of drugs. And we've subsequently gone on and we've sequenced the genome of these strains, and we now know exactly what has changed in the bacterium and how the protein has changed to be able to pump out these drugs to a greater level. So now we know how this works, we can now understand how to start inhibiting this pump and starting to develop inhibitors that you give at the same time as the antibiotic to then restore the activity. So I've just told you a little tale about salmonella. What else are we worried about? Well, this is just a hit list of different bacterial species within the hospital or within the community that have antibiotic resistances that make them very difficult to treat. Now, we used to always talk about hospital-acquired pathogens and community-acquired pathogens. The reality is nowadays they're getting blurred, and whilst we used to make the distinction, this is happening less and less. So we can have patients in community with ESBL type of resistance, shown here with the third bullet on the left hand side, and likewise we can have patients who will have TB infections or MRSA infections that will go into the hospital. So this is another heat map saying, well, how much resistance is there really? I've just shown you a couple of examples. Is it widespread or is it something that is rare? So if you look on the left hand side here, this is the prevalence of invasive Klebsiella. So these are bacteria that are sort of the sisters to E. coli. They're in the same family as E. coli, Klebsiella. Invasive just means that they go into the bloodstream. So again, these are bloodstream infections. And you can see, just like there was with the consumption heat map, there are higher numbers of antibiotic-resistant bacteria in southern European countries than north. And on the left-hand side, these third-generation cephalosporins, these were developed to be active against penicillin-resistant bacteria. So these started being used in the late 1980s, 1990s. Now we've got a problem with a lot of resistance around to these. We're now using drugs called carbapenems. But on the right-hand side, we can see we've already got resistances emerging and being fairly commonplace in some countries. So we use one drug a lot, it becomes ineffective, we move on to using another one. And then we use that a lot, and that becomes ineffective, we need another one. And we're running out of options. So resistance really is global. I've shown you European figures because we're very good in Europe at keeping these figures, less good at keeping figures for elsewhere. And this is just an example of the resistances that Klebsiella has got to the drugs shown on the right-hand side of the previous graph, the picture, the carbapenems. So this is a type of resistance called KPC, and a K means Klebsiella. And the take-home message I want you to get here is that some of these resistances emerged quite a long time ago. So if we look here at the United States, this resistance was first reported in 1996, but it was considered to be very rare and not likely to spread anywhere. But what we've got now is these enzymes are appearing all over the world, and that's what's giving resistance to these drugs. Now, how is everything going all over the world? Well, we're taking these things all over the world. They're not emerging individually, spontaneously, all in parallel. Of course, there have been separate events occurring, but we share our bacteria. And if we look at the picture in the middle, most of us live in urban areas. We mix very closely, and we are sharing our bacteria, the same way as we share our viruses. We don't live in a sterile world. We don't want to live in a sterile world. We have the microbiome, the biology within and on the surface of our bodies. Those have very important roles to play. But we're also sharing the pathogens as well. Now, for instance, if one of you had gone to the bathroom before you came into this lecture, bit of an upset tummy, rushed to the loo, were rushing in here, didn't wash your hands that well, you came in and touched the plate there, the bacteria that were on the hands were on the plate. The next person in touches that plate, they're on their hands. That person who's just touched the plate and got in their hands, they didn't have time for dinner before they came out, so they've got a sandwich in the bag. They're sitting in the back there because they think I can't see them. They're going to eat the sandwich. The bacteria that were on my hands, on the plate, on their hands, are now on the sandwich. Now they're eating the sandwich. Now they're in their tummies. Okay? That's how easy it is to share our bugs. That's how we share them and take them all around the world. We also know about keeping things clean in hospital. We do this because we know that patients are more vulnerable. So we understand about infection control. We just don't relate it to ourselves when we're outside of hospitals. So we share our environment with other people, the other people that we live at home with. We share the environment with our pets. So we're giving them our bacteria, they're giving us theirs. With farm animals, we eat food that's come from farmed animals. The sewage gets sprayed on fields in some countries from animals and sometimes from people. So sometimes salads are covered in sewage in some countries. A lovely story, isn't it? We transport our food everywhere, and food can have bacteria on it. So we're living in a sea, a whole world of bacteria. So what does this really mean in terms of resistance? Well, a study was done recently on Swedish people when they were returning from their holidays. They weren't ill, they were just screened in a volunteer study to say, what are you carrying in your tummy now you've come back from your holiday? They were screened before they went on holiday too. A quarter of them had that ESBL type resistance that gives resistance to cephalosporins, which they brought home with them from their holidays. So we are taking bacteria with us, and we're taking antibiotic resistances with us. Of course, not everybody has the same luxury of the high standards of living that we have. This is how many people live in different parts of the world. So they are exposed continually to antibiotic-resistant bacteria, as well as other microorganisms. This will be their water source as well as their sewage system. And in countries such as this, these are the very people who are able to buy antibiotics over the counter and start pushing the selection of antibiotic-resistant bacteria. So we can select them when we're exposed to antibiotics. In this environment, they'll use even more antibiotics, making an even greater pressure. I'd like to now talk just a little bit about antibiotics in animals. So this is an example of how much antibiotic is used in animals in the EU. And straight away you can see that some countries, including the UK, seem to use a lot of antibiotic, and other countries use very little. One of the reasons for this is based on farming. Certain countries within Europe are very big food producers. They export a lot of animals that are used in food, and so they use a lot of antibiotics as part of the food production process. So if there is a country on here, such as Iceland, that doesn't have a lot of pig production, they're not going to use a lot of antibiotics that are typically used in pigs. So these numbers can appear a bit distorted. Out with that, you can see quite clearly there's an awful lot of antibiotic used in animals. Fish production, they use a lot of antibiotic in fish production. So if you think about salmon farming, those are in compounds, in inland seas and waters, coastal waters. Just pour the antibiotics into the water. China is the biggest fish producer in the world. In 2008, as you can see, the figures there are absolutely massive. Over 32 million tonnes of fish a year. They're also able to have an awful lot of antibiotic resistance in their fish. So a colleague of mine, Peter Hawking, who works here at the university, when he was doing some work with collaborators in China, they just bought some fish off the local market. They took them back to the lab, they grew up the bacteria, and they just took a single E. coli from each fish, and again we've got these cephalosporin resistant, these ESBL resistant bacteria in these fish, so people are eating them. And CTXM14 is an enzyme that I'll talk about in just a little bit more detail. First, I want to talk about how do we actually give antibiotics to animals. So we first of all are probably familiar with the phrase growth promotion. It's been in the news quite a lot in the last 6 months. And this is something that happens outside of the EU. It was banned in the EU in 2006, but quite a lot of countries, including the UK, had a voluntary withdrawal several years before. This is where antibiotics were given to animals with the sole purpose of making them grow bigger, faster. It was not to treat an infection. Outside of the EU, they still do this, and it's a very widespread practice, although it sometimes masquerades under the heading prophylaxis. Anyone here know what prophylaxis is? Anyone like to shout out what they think prophylaxis is? Okay. So a couple of you know, but not everybody. So prophylaxis is where, for instance, you think there's an infection and you don't want it to spread to any of the other animals around, so you'll dose all of the animals. Now actually we used to do this to people as well, and we still do in certain situations, but before we had the meningitis vaccine, which most of you in this room have had. We used to have sporadic outbreaks of meningitis, including in university halls of residence. And indeed at Birmingham we had a couple of outbreaks. So after a couple of the students had gone down with meningitis, every other student in that hall of residence would have received an antibiotic as prophylaxis. In other words, as a preventative measure to stop them going down with meningitis. So some places will still use antibiotics in animals as prophylaxis, even though there is no particular infection that's been diagnosed, but they want to prevent infection. Why do they want to do this? If you've got a flock of 20,000 birds, say poultry, and they start to cough, you do not want to risk the huge costs of losing that entire flock, so they administer antibiotics to the entire flock. We do use antibiotics for treatment in animals as well, so racehorse is very valuable. They're treated on an individual basis, and of course our pets get antibiotics too. This is the sort of difference in the amount of antibiotic that's used in the USA in animals compared to people. Over 3 times more by weight is used in animals. The big problem with using antibiotics in animals for food production is that they're treated en masse, whereas when you get an antibiotic, only you get it, everybody else in your house doesn't get it, or in your class at school. So imagine that you've got an antibiotic, but everybody else in your school now has to have it as well. That's the situation with animals. The worst part is that they're not going to get the right dose. They're not even going to get it at breakfast, lunch and dinner. They're going to get it in their food or their water, and they're allowed to drink or eat as much or as little as they want. So an awful lot of them are going to be underdosed, and that's almost the perfect way to make an environment for little stepwise changes in susceptibility to antibiotics. So little by little the bacteria become less and less susceptible, and the antibiotics become less and less effective. So this is a study we did a few years ago, and it's on a bacterium called Campylobacter, and this gives food poisoning. It's the most common cause of bacterial gastroenteritis. So it's much more common than Salmonella, although most people think Salmonella is the most common cause. And fluoroquinolones, of which ciprofloxacin is a fluoroquinolone type of drug, were introduced for use in animals in the late 1980s. And we monitored the number of people getting Campylobacter infections that were also resistant to this antibiotic. And there was a great association with the introduction of resistance and an increasing resistance. Now, the same was shown in other countries. What we do know, and I'm sure you know yourselves, when you get an upset tummy, how many of you have ever been to the doctor with an upset tummy? Okay, how many of you have had an upset tummy? Okay, so that was quite interesting, because I can tell you now, only about 10% of people with an upset tummy are so ill they go to the doctor. And they've usually waited 3 or 4 days, and they're usually pretty ill by the time they get to the doctor. Which means that we're only being able to grow about 10% of the bacteria that are actually out there causing disease. So we've got at least 500,000 bacteria infections. We also know our current rate is about 15% Campylobacter resistant to Cipro. That's an awful lot of resistant infections. So we and others said, well, these must be coming from via the food, because that's where the drug's being used. We're not using this drug really in people, so it's unlikely that they're going to be getting Campylobacter that are resistant any other way, because Campylobacter isn't a commensal. If you've got a Campylobacter in your tummy, you are ill. Otherwise, it's not going to be there. So it's not like E. coli. You're either ill or you haven't got it. So we did a study with a group from the University of Bristol and a national reference lab, where we actually had the agreement of farmers to go in and sample their poultry, both before, during and after treatment with a fluoroquinolone. And we then analysed both what we got out of the birds for the Campylobacter, their antibiotic resistance, and the type of resistance. And as you can see very clearly here, shown in the dark bars, Once treatment started, the very rare bird with the dark bar which shows resistance became, all of them had resistance, so they'd all shared their bacteria. These bacteria persisted, and they were there until those birds were slaughtered and entered the food chain. We did the molecular biology on these bacteria, and the resistances were identical to those we'd seen in the isolates from people. There is, however, a word of warning that what is true for one antibiotic is not necessarily true for all antibiotics. And in the top here, we show a flock of birds before and after treatment with chlortetracycline, which is the sister drug to Oxytet that some of you might have used on your skin. And in the top, you can see there is a lot of resistance in the untreated flock and a lot of resistance in the treated flock, so it didn't make any difference. The bottom, we had a few resistant strains around in a few of the birds, and they didn't become the dominant population, so very different to ciproxin. So I'm going to change tack again now. Up until now I've been talking predominantly, and particularly I've talked about ciproxin resistance, about resistance that's emerged through evolution and mutations on the bacterial chromosome. Bacteria are really good actually at having sex too, and this is what's shown on this slide. So, oh no, it was really embarrassing, but they do, and they're really good at spreading things around. So, if you look at the left-hand side of this spiky bacterium here, there is a little tube going all the way to another bacterium. Okay, shown here. This bacterium here is transferring down this tube, this sex pillar, DNA that is outside of the chromosome, extra chromosomal DNA that we call a plasmid. And on it will have antibiotic resistance genes. It might also have genes that give toxins. So E. coli 0157 has acquired the toxin by bacterial sex. Now, bacteria really like having sex. They have a lot of it, and they have a lot of it with other species. So it's not always an E. coli to an E. coli. It might be an E. coli to a Klebsiella. This is just a diagram here to show you how this can happen. The other important point is that not only can they transfer resistance genes this way, that evolution to those resistance genes can happen as well. So mutations don't happen just in chromosomal DNA, they will happen in any bit of DNA. It's a random, spontaneous event. So we could have had a resistance gene that was transferred between bacteria, and then we've developed third-generation cephalosporins to be active against that. And then that gene has acquired a mutation to be able to get around the third-generation cephalosporin and survive. So CTXN14 is one of the resistances that I mentioned to you that were in those fish from China, and this resistance is throughout the world, as shown through this map here. It's not the only form of resistance, but it's the second most common worldwide. So in 2006 was the first report of this type of resistance in animals, and it just happened to be a report from the United Kingdom, from a calf in North Wales, and further investigation by the Department for the Environment, Food and Rural Affairs, DEFRA, showed that actually there was quite a lot of it about on this farm. So at that time I became involved in this study, and what we did is that little bit of extra DNA that's transferred by bacterial sex, we sequenced the entire bit of DNA, so the plasmid genome. And what we then did is we then designed a test that we could rapidly screen the bacteria with these plasmids. So basically what we then did is once we had the bacterial strain, we put it in a little tiny test tube, we boiled it at 100 for a few minutes. That's enough to spill the DNA, kill the bacteria, and then we used something called the polymerase chain reaction, a PCR. The photocopier that we use in the lab got hundreds of copies up, and we were able to see this. And to cut a long story short, we were able to show, by getting strains from all over the world, that this little element that was found in a calf in North Wales was identical to one that had previously been described in 1998 from a patient in China. How on earth did it get from China to North Wales? And then we found that this element was all over Spain, whether the bacteria came from the environment, from animals or people, and something very similar was found in Australia. So we then did additional tests, and it didn't matter where we looked, and this is just some UK data, it doesn't matter if they're animal isolates or human isolates, this little element is everywhere. So then we said, well, it must be the antibiotic that's been driving it. So we did some genetic manipulation, and what we did is we took out the antibiotic resistance gene from this little element, and we did experiments with antibiotics to say, well, this is going to have to disappear now, it's only going to be favourable in the presence of antibiotics. And what we found is, in fact, yes, it's favourable in the presence of antibiotics, but this element is not a burden on the bacterium. It's obviously doing something else. It's obviously giving a survival advantage in something else, another environment. We don't know what it is, we're still trying to work it out. So what we don't know now is whether this element went worldwide first and then the antibiotic resistance gene went into it on several occasions, or whether the antibiotic resistance gene went into it, got spread worldwide, and then when we chose to use the antibiotics, we made the situation worse. And this is the sort of experiments my lab are currently grappling with. Which came first, and how are we now going to defeat these? So what we'd like to do is discover ways to stop these little bits of DNA transferring from bacterium to bacterium, and we're devising new tests to do this. So let's go back to resistance and outcome. So we've got a lot of resistance around, but how often does it really cause a problem in patients? Well, we know there's a really, really good correlation between patients with infections in their blood who are very seriously ill and very poor outcomes. So more of those are going to die because we can't treat them. Many of those patients will be on intensive care units, ICU. So there's, again, a good correlation between antibiotic resistance and poor outcome. And there's also a good correlation if they happen to be infected with these bacteria listed here. Although there's a less good correlation for infections outside of the bloodstream, or things like Streptococcus pneumoniae that cause lung infections, what that really means is we don't have enough evidence to say one way or the other yet. My personal view is that there will be a good correlation, because wherever we look and we do really well-designed studies, we find the association that antibiotic resistance means it's a really bad news for patients. So, okay, some patients are going to die, but what does that matter for the rest of us? You know, let's be really hard-nosed about it. People have always died of infections for thousands of years. What it actually means for us is that if we had that antibiotic-resistant Campylobacter from eating poultry, we might not have been ill enough to go to the doctor, but what would have happened is we would have been ill for longer. And I have actually had an antibiotic-resistant Campylobacter infection, and I was off work for 3.5 weeks. I really thought I was going to die. It was terrible. Okay? So this is the impact. It's beyond just the antibiotic resistance. These resistances change the bacterium as well. They make them sometimes more virulent and last longer. Of course, there are real costs to society as well. What this means is that it costs more. So it costs longer because you're in hospital for longer. It costs more because you need more drugs. It costs more because I'm off work and somebody's got to do my job when I'm not at work. You're off school, which means you've got to catch up. So there's all sorts of costs. We can measure these financially. If you measure it for Europe, and this is data from 2010, and it was not a massive study, but by extrapolation, they think there were at least 400,000 additional infections, giving an extra 25,000 deaths. 2.5 million extra hospital days, and the cost to the EU was €1.5 billion. Similar, very high costs in the USA. This led to the World Economic Forum, when they met in Davos in Switzerland earlier this year, putting antibiotic resistance on the global risks register. This is one of the figures from this. This is freely available should you actually want to look at this document. It's very readable, and for the teachers that are here, this is a really good document. It gives you a very good up-to-date picture. I'm just going to draw your attention to one point on here. This sub-Saharan Africa, Tanzania. Everyone's heard about malaria and how dreadful this is in low-income countries. In children, antibiotic-resistant bacteria gave them twice the death rate to that of malaria. So this is a massive problem all over the world. We are in a vicious cycle of antibiotic resistance. The more resistant strains, the more antibiotic we use. The more resistance there is, the more antibiotic resistance there is. And we're increasingly using more because of this cycle. But we have fewer and fewer agents. If you look at this timeline, you can see the number of new drugs reaching market has absolutely plummeted since 1995. Part of this is many of the easy, and I use this very reservedly, easy to develop agents have all been used up. Easy, many of the antibiotic compounds that we find in laboratories or from natural products never make it to a medicine. It takes 5 to 10 to 15 years to go from a discovery in a laboratory to a drug in a patient. It's never easy, but it's absolutely drying up. There have been no new antibiotic classes discovered since 1987. One could argue, well, does that matter because we've still got lots of targets? But what it means is we're always doing a new type of penicillin, for instance, and ultimately we'll run out of new types of penicillin, so we will need new targets. So why aren't there any new antibiotics? There's lots of reasons. Big companies say they're not making enough money from it. There's a depletion in skills. Not everybody is doing this anymore, particularly big companies. Now they're not doing this type of research. The golden age of discovery, many of the people involved have now retired, so we're actually losing research capacity. So labs like mine are pretty rare, where we've got a big lab we've been running for many years, and so we need to increase capacity. And there's the other issue that clinical trials are really hard to do for antibiotics. So if I give you an example of breast cancer, if we've got a new drug we're trying out for breast cancer, every person going into that trial, you know they've got breast cancer. You've had several weeks to do an accurate diagnosis. If a patient has a bacterial infection, you've got 24, 48 hours maximum to get them on an antibiotic. Usually, if they're in hospital and they're seriously ill, you've got hours. We cannot diagnose what species is causing an infection in under a day. It's very rare. We still use Victorian technology. We grow them on an agar plate, and then we do susceptibility testing, often on an agar plate, although we can do some tests more rapidly. And what this means is we have to put patients into clinical trials blind. They may have a chest infection. We're trying out a new drug for chest infections, but we're aiming that drug at a particular species, such as Streptococcus pneumoniae, but we wouldn't actually know the patients had got that. So we end up enrolling thousands of patients to get 100 who've actually got the infection we're trying our new drug for. This costs millions to do this. So number 1 and 2 drug are not antibiotics, but look, $5,000 million per year in profits. That's what companies want to make. Antibiotics, numbers 21 and 138 on this scale, they're still making millions, but as you can see, the second top selling antibiotic is now only making hundreds of millions. And any new antibiotic is likely to make tens of millions. And what companies want, they want their blockbusters, they want their thousands of millions of dollars profit a year. And if it's costing them much, much more to develop antibiotics than other therapeutic drugs, the commercial sector is always going to go for another option. There is cautious optimism, certainly in discovery. There are quite a lot of labs now doing lots of innovative things. There's phages, which are viruses that infect bacteria. In fact, even on Friday, headline news, University of Leicester, they've got a bacteriophage against Clostridia, and they've just sold it or licensed it to a company to develop. Unfortunately, that will still have to go through the same regulatory hurdles as an antibiotic. So we're again still another 5 or 10 years away, even if they get a product useful for people. We've got a bacteriophage to use against salmonella, but it's getting the take-up from the commercial sector to develop these interesting products. We can manipulate bacteria to make them make antibiotics. Now we can genome sequence and we can see they can make lots of wonderful things. But it's translating that and getting people to turn that into a product. So this is what's led to the global concern. Our Chief Medical Officer in the United Kingdom, Dame Sally Davies, has been a massive advocate for this. She's in fact got a book out. It came out 2 weeks ago called The Drugs Don't Work. You can get it via Kindle. It's about £1.99. It's very readable. We have to start tackling this problem, because if we don't understand resistance and understand how we ourselves are all involved, we're not going to be able to have antibiotics for the future. So we need to start recognising that antibiotic resistance is affecting millions of people in the same way as people were frightened of catching AIDS in the 1980s and 1990s, and this stimulated all the anti-HIV drugs, but this actually also stimulated lots of other antivirals to be made. We need to get the same global concern now for making new antibiotics. But really importantly, we've got to preserve what we've got now, because we don't know when we're going to get these new drugs. So we've got to be really careful about what we've got. Department of Health has something called Start Smart Then Focus, and all that means is think very carefully about what you are asking for as patients, and doctors have to think very carefully about what they're giving. And we need to find new treatments. We need desperately to do lots more research. 18th of November every year is European Antibiotic Awareness Day. Anybody here ever heard of that before? Hands up. No. You've heard about it now. Oh, well done at the back there. So, you've heard about it now. Promoting the responsible use of antibiotics. This is really important, because the people who are most likely to go to doctors for healthcare are young adults, and they often ask for antibiotics. Because as children, you went to the doctors with usually your mother, but you went with a parent, you had an antibiotic and you associate it with getting better. Often you would have got better anyway, particularly if it was a cold or flu. That's a virus, an antibiotic's never going to work. So this is the way doctors often will prescribe antibiotics. It's a leap of faith. So they're diagnosing what they think is the most likely infection, and therefore the most likely drug that's going to be effective. What we want to move towards is the targeted approach, where we can diagnose infection much better than we can now and give an antibiotic when needed. The best case at the moment is that we're going to start being able to diagnose much more rapidly a virus, and then know, well, it's a virus, therefore it isn't a bacterial infection, and you've got flu, for instance, therefore you don't need an antibiotic. So antibiotic action, some of you probably saw the banner outside, is a public awareness initiative to make everybody aware of the problems of resistance, because if everybody becomes aware of it and everybody tries to help resolve the problem, then we will have treatments for the future. So when I need my hip replacement, or someone out here unfortunately will need a kidney transplant, they get an infection, they'll be able to treat it. We launched this initiative at the House of Parliament just under 2 years ago, and it's had an amazing effect, both in this country and abroad. We took a petition to Downing Street, work very closely with politicians both in this country and abroad. I'm actually director of this initiative, so I'm 80% University of Birmingham, 20% time for this initiative. We're working with colleagues throughout Europe and in other countries, and we have an all-party parliamentary group in the United Kingdom, which means politicians of all parties are now involved and recognise this is very serious. And our Department of Health has an antimicrobial resistance strategy. It was published a couple of weeks ago, and we'll have an action plan in the new year. So we're really signed up in this country. And Dame Sally Davies, our Chief Medical Officer, not only is she a great advocate to us, and she's appeared on television a lot, she's taken a lead on a global stage, and in fact this week has been chairing a WHO meeting. So everybody's really signing up now to recognising that this is a problem. So this is my last slide. I'd really like you to help us spread awareness about this. The Antibiotic Action website is also a great resource for you for your A-levels. There's an awful lot of information on there about resistance. But please sign the petition and spread the word. Thank you very much, everybody.